Hello, I'm Dr. Marc Rothenberg here at Cincinnati Children's Hospital. And what I'd like to do today is share with you new data that we have just released about an important study concerning eosinophil depletion in eosinophilic gastritis. This is an important study because eosinophil-associated GI diseases, or EGID, have a name because eosinophils accumulate at high levels in the tissue. There's been a strong focus on the eosinophil in this disease. Certainly, eosinophil levels are high. Eosinophils are known to be toxic cells. If you read most articles about eosinophils, when you look on the internet, you'll find that eosinophils have been implicated as actually the causal cell for these diseases. Yet, whether or not this is true was uncertain. In this study, we tested the effect of an eosinophil depleting antibody called Benralizumab on the outcome in patients with eosinophilic gastritis. Some of these patients had concurrent eosinophilic esophagitis and duodenitis. This was a single-site study conducted at Cincinnati Children's Hospital, and we examined 24 patients, half of which underwent placebo and half which underwent drug exposure. As you can see in this diagram, patients underwent a screening period. They then had a double-blind, placebo-controlled phase of this study, which was conducted over the course of 12 weeks. During this time, patients received 3 injections of either Benralizumab at 30 milligrams or placebo. No one knew what the patients were getting. At the end of the 12 weeks, the patients underwent an endoscopy. After these 12 weeks, the patients were entered into 2 different long-term open-label extension trials that extended up to a total of 88 weeks, which is quite a long time for a clinical study. This ensured that the patients were exposed to the medication, had eosinophil depletion over time. One can see in the bottom panel that the drug was indeed very effective at reducing eosinophil levels. Many of the patients had no eosinophils detected, which is common with this drug. In contrast, placebo patients typically maintain their high level of eosinophils. Similar findings were in the blood, where patients had a near loss, a complete loss of eosinophils, many of the patients with the drug. Now, despite these findings, which was eosinophil depletion, we found that the features of the disease, including endoscopy, histology, symptoms, including pain and non-pain measurements, were unabated by the eosinophil depletion. That's shown in these graphs here. They're complicated slides, but basically the lines are not showing a consistent decrease in these parameters. Now, the histology or the changes in the microscope do improve, but those features were limited to only those associated with the eosinophil decline. During the open label period, there was a modest reduction in pain, but please keep in mind that this was considered modest, and this is an open label study. I think what's most forthcoming and informative is that we also took biopsies from the tissue in these patients, and we subjected them to a unbiased, completely objective analysis of molecular biomarkers looking at a panel of genes. And what you can see here in the right panel is that the patients all look the same, and they look different from the controls on the left, which was the non-eosinophilic gastritis. Now, when I say they all look the same, you can see that these 48 genes that represent many different features of the disease were similar between the patients that were the controls who had active disease, the patients at baseline, and following the placebo-controlled trial, the 12 weeks for both the Benralizumab and the placebo. Now, there were changes in 2 genes, and those 2 genes were eosinophil markers called CLC and CCR3. That indicates that indeed the molecular analysis is working. But what's notable is that the disease features were really not improved by any of these parameters. These findings really call attention to the role of eosinophils and suggest that these cells aren't really the culprit. These collective findings indicate that the pathogenesis of EGID probably involves coexisting eosinophil-independent pathogenic mechanisms, and that successful management might require therapies that inhibit pathways that more broadly reduce type 2 inflammation rather than only targeting eosinophils. Furthermore, our results suggest that defining disease remission in EGID by eosinophil counts alone should be reconsidered, given that core disease features persist despite eosinophil depletion. Before closing, I want to thank so many people that have contributed to this, especially the patients, the clinicians that were involved, the funding agency, which was primarily a partnership with the drug company that makes the Benralizumab, AstraZeneca, and the team at large at Cincinnati Children's that conducted this study, especially because it was hard to recruit patients for this rare disease, and this was a single-site study that was conducted in a very elegant fashion by the research team. I'm very grateful for the patients that had faith in us, and I want to let you know, although these results were unexpected, they really advanced the field because they now tell us that we need to shift our attention away from eosinophils, and indeed our research operation here at Cincinnati Children's already has a number of studies underway looking at other approaches and targeting other cells and other pathways that we think are very important in this disease. So the optimism is high, progress is being made, and again, I thank all of you for your interest in this and partnership. Have a nice day.